So I'm Vanessa. I'm a fifth-year graduate student in Darren Moore's lab here at the Institute. And Darren's lab primarily focuses on studying genetic forms of Parkinson's disease. And most of you are going to have a sporadic form or a non-genetic form, but about 10 to 15% are going to have some kind of genetic contribution. We're really focusing on what that 10 to 15% can tell us about Parkinson's disease. All right. Everything's working here. So I'm going to give a little bit of background. So Parkinson's disease is a disease of the brain. It happens in a specific pathway in the brain degenerating over time. So we have four major dopamine-producing pathways in the brain. The one we're interested in is this nigrostriatal pathway right here. So these are dopamine-producing neurons or dopaminergic neurons that project from the substantia nigra, SN, to the striatum or STR, and they're involved in sort of movement. And so dopamine overall is a neurotransmitter, and when it signals in this pathway, it's responsible for movement, but other signaling, it can be involved in sort of motivation and reward. So it does multiple things in the brain. This pathway exists both in the human brain and in animals. We can use it in animal models to study sort of what happens in disease. And we want to do this because understanding how these neurons are dying can help us identify potential ways to intervene. And summarize, Parkinson's disease, nigrostriatal pathway degenerates. When we look at the nigrostriatal pathway in the brain, we can identify how these neurons are dying, can prevent them hopefully from dying in Parkinson's disease. So genetics in Parkinson's. So like I mentioned, a subset of Parkinson's disease is going to be caused by genetic factors. So this is a graph showing us the genetic landscape of Parkinson's. So along the bottom here, we have how frequently certain genetic variants occur in the population, from incredibly rare to very common. And then in this vertical axis, we're seeing how much of a risk for Parkinson's disease does having one of those variants give you, from very low risk to very high risk. So we can appreciate that there are three main groups here. In green, we have very common but very low risk variants. So having a few of these may very slightly increase your risk of developing Parkinson's. In the middle here, some of these factors look maybe familiar. So these are uncommon but slightly higher risk variants. These represent the most common genetic drivers of Parkinson's disease. So most people who have a genetic component will have either a GBA or maybe a LRRK2 mutation, but these are not always causal. So having one of these variants doesn't mean you necessarily will get Parkinson's disease. And then finally, this interesting cluster at the top, these are exceptionally rare. They occur in a few families around the world, but they have very, very high risk for Parkinson's disease. So these are considered single genetic mutations that will drive PD. And so these are about 5 to 10% of PD cases. So do these mutations tell us anything? Well, this group of mutations that we just saw in pink all kind of converge on the endolysosomal pathway in PD. What is the endolysosomal pathway? Essentially, if you took a post office and a recycling plant and combined them, you'd get the endolysosomal system in cells. So this is a system in which a cell can take its proteins and can send it through this central shipping hub where it can be trafficked to where it needs to go, delivered to different locations in the cell. It can be recycled, reincorporated into different areas and reused, or if it's damaged or degraded or no longer needed, it can be thrown away. So this is a picture more accurately of what this looks like in the cell. So here's a cell with the nucleus. We have a couple different bodies here. These are organelles. And then you can see some of these pathways like LRRK2, TMM230, VPS35. These mutations all have a role in this system. So they're all doing something within the endolysosomal pathway, which tells us that this pathway is probably pretty important for Parkinson's disease because when you get a mutation in it, you get the disease. And so we want to know, what can these mutations tell us about how the endolysosomal system plays a role to Parkinson's disease? So in order to study these mutations, we first need to identify them. So how do we find out what mutations are causing disease? So essentially, we do something called whole gene sequencing, which is kind of like a very long spell check. So we take individuals who have familial Parkinson's disease and we take their DNA and we align it in comparison to families, so related family persons who do not have Parkinson's disease. And then we look to compare the differences in the genetic sequence between these groups of people to see if there's a pattern that we can pull out. So for example, in this example, we have a couple differences here in some of the persons with Parkinson's, but they're not consistent among the patients. So they're probably not driving the disease. In this example, we have some mutations, but they're not tracking with disease because we see it in healthy persons as well. It's probably not driving disease. So we keep going until we find something that looks like this. The proposed variant is in all of the persons who have Parkinson's disease, but it's not in any of the unaffected family members. And so this tells us, because it's tracking with disease, that it's probably the variant that is driving that disease. And so this is the process that we did for, or not that I did, but that a group of researchers did for a family that was in Canada. There were about 81 people in this family, and across multiple generations, there were people presenting with inherited Parkinson's disease. So this is a genetic pedigree, which is kind of how we track inheritance. Each of these little diamonds represents a person. When the diamond is filled in dark black like this, it means that person has Parkinson's disease. And so they went through and collected DNA from a lot of these people, both the affected members and the unaffected members. And when they did that sort of spell check, they found that there was a mutation in a gene called transmembrane protein 230, or TMEM230. So it's a gene we don't really know a lot about, but when they went and looked in international databases to look for other families with Parkinson's disease, they found mutations in TM230 as well. So not only was it in this one family, it was in a few other families around the world. So that leads us to ask, okay, are TM230 mutations causing Parkinson's disease, and how are they doing it? So to understand how they're doing it, we need to make a model of TM230 linked Parkinson's disease in the laboratory. So we need to take these mutations and see if we can create models of them to study how they affect the cells. And so to do this, we're going to use some viral vector technology. So we heard a little bit about this in the gene therapy segment, but basically we can take a virus, we can empty out all those components, and I can put in my sort of gene of interest. So I can make a vector that's ready to deliver TM230 to cells. So it'll integrate into the cells and let them produce that TM230 protein. And so we take this, we add it to cell culture, and we can add it to animals. When we add it to animals, we add it, or at least I chose to add it in a one side specific manner. So we have this little brain section here. I've added it to one side to create an affected half of the brain and to leave one half of the brain unaffected. This allows us to directly compare two halves of the brain and to see what changes in the affected side and what stays the same in the unaffected side. So it's an internal control for us. And then to briefly kind of go over how these vectors work, again, we put them in the cells, they enter the nucleus, they release that DNA that I've encoded into them, into the nucleus. That DNA gets turned into instructions for building proteins. So there's that mRNA that Tommy spoke of a little bit. That mRNA leaves the nucleus, and then we have sort of the assembly machinery found in the cell, which takes those instructions and builds the proteins based on those instructions, which causes the protein of interest to be produced in those cells. Now, as part of this process, when I create these genes, I add a little tag to the end of them so that when I make this protein is built with a little tag at the end of it, and that tag allows me to specifically track those proteins as they move through the cell. So it lets me visualize what proteins I've put into the cells, which then lets me ask questions about those proteins. So we can ask, for example, well, where does it go when it's in the cell, right? Where does this protein naturally track to? What happens to the cell? And what causes that effect to happen? So do the cells die? And if they do, why do they die? And of course, because every good science experiment starts with a hypothesis, we hypothesized that overexpression of wild type or natural normal mutant or normal TM230, as well as the mutant versions of TM230, would likely lead to cell death or neurodegeneration. Okay, and so we'll look at this picture here, and anyone can stop me anytime for questions. Yes.
Previous slide.
Yes.
What's the time frame for that process to take place?
Right, that is an excellent question. So we inject the virus into the brain. The virus will integrate into cells and probably start producing protein within 24 hours, and then the protein can continue to be produced up to a year after we've added the virus. So it's a much faster model than some genetic models where we mutate the host DNA and then let that change. But most viruses, we keep the animals for up to, say, 12 weeks and really can see effects starting as early as 6 to 12 weeks. So it's a relatively quick process. So here is some images. So these are images of staining of the brain. And so we have two sets here. So remember, we had an unaffected side, which is this stack at the top, where I did not add any of that vector expressing TMEM230. And we had an affected side at the bottom where I did. And so the first thing we just have to do is check that that tag that I mentioned is tracking the protein to make sure that we can actually see and follow where it's going in the brain. And so you can see V5 here is that tag. TMEM230 is the protein of interest. Where I didn't put any TMEM in the brain, we don't see any TMEM, which is a good thing. So it's not where I didn't put it. Where I did put TMEM230 in the brain, we can see it, and we can see that the tag follows it exactly, which means that that tag is accurately picking up that protein. From there, we can look a little bit more big picture and see where exactly that protein ended up. So these are whole brain slices of the midbrain region. So this area here is the substantia nigra, which is where those dopamine neurons originate. And we can see that when I inject it into one side, we see that nice brown staining localizing to the substantia nigra of that side. So we know that it is going to the area that we want. And we can see when we look close up that these little circles that the brown seems to be filling up look an awful lot like neurons. So we think it is hitting those dopamine neurons that we want to see. Now we can kind of start to ask what's happening to those neurons now that we've delivered that protein to them. So one of the other questions we had, obviously, is where does it exactly go? And this is something you can answer by co-localizing the markers. So basically, when we stain for V5 to track the protein, we can also stain for a different marker, marking another location in the cell to see if that protein ends up right next to it, right? So we're trying to see if physically these two markers overlap. And the answer is that they do. So GM130 is a marker of the Golgi. So the Golgi is kind of the shipping hub of the cell. It's part of that endolysosomal system. And when we stain for the Golgi, we can see that the green ends up right next to it. So this is a little bit hard to see in this corner, but you see the yellow there. So when red and green overlap, we're going to see yellow. And that tells us that this protein is ending up in the Golgi, which gives us a little bit of a clue that the function it's having probably is related to what the Golgi does. So why do we care about the Golgi, right? So the Golgi is part of that endolysosomal system. It essentially is the shipping center, right? So the cell can take its proteins. It can say that it wants to deliver them to a particular location. The Golgi packages them in little vesicles. It directs them to where they need to go, and they get delivered to that location. Cell trafficking. It's a really important, essential part of the cell maintaining a healthy function, right? However, in neurodegenerative diseases like Parkinson's disease, we actually can appreciate that the Golgi starts to fragment. So that the Golgi, which is this nice kind of stacked long ribbon structure, will actually start to break apart and physically dissociate. And when that happens, it loses its ability to effectively package and send these proteins where they need to go. And so we get disrupted cell trafficking, which can cause a cell essentially to dysregulate, dysfunction, and could lead to cell death. So this is something we don't want to have happening in our cells. So here we have a picture of two primary neurons in cell culture. So these are rat neurons that are growing in culture. And I have transfected, or I've added that mutant version of TMM230 to this culture and to have that producing in those cells. And that's stained in green here. And so in this one neuron down here, if I blow this up, if we look at that TMM230 staining, we can see that it's got this nice sort of consistent ribbon-like structure. So it looks very connected and very normal. And so this kind of tracks with what we would expect a healthy, normal Golgi to look like. But in this same culture, in this second neuron up here, this is the same kind of staining, the same TM230, but it looks very different, right? So all of a sudden, it looks very broken apart, very disjointed. We have this kind of blobby type pattern. And so this looks almost like it's tracking with a fragmented or an unhealthy Golgi. And so there's early evidence here that perhaps something in the Golgi is being dysregulated when we introduce that TM230 mutation. Now, we haven't confirmed this, but we are actively doing more studies to kind of try to see if we can pull out this pattern of when we put the mutant TM230 in there, does the Golgi start to break apart? So beyond that, we're also looking at the dopamine neurons overall, because another important measure is just overall cell death. When we add the mutant TM230 to the animals, do the neurons die? And so we can look at this with this heat map. So what we're looking at is several groups across here. We have a control group at the top, which should be healthy normal animals, and then a normal sort of unmodified TM230, and then 3 different conditions where there are different mutations of TM230 that were found in PD patients. This is striatum of brains that were stained for tyrosine hydroxylase, or TH. TH is just a marker for the dopamine neuron. So the more TH there is, the healthier and the more neurons there are. So this little guide at the bottom will tell you that where it's red, we have a lot of those neurons, and where it's yellow, we have much fewer of those neurons. And so what we're looking for is we're looking for examples. I think this animal here is the more obvious one, where in the affected side where I put the TMM230, are there less neurons than the unaffected side? So I think in this one, you can see pretty clearly we have a lot more yellow here. There are relatively fewer TH neurons than there are in this side. So it looks like where the TMM230 went, those neurons died, and we got a reduction. I can quantify this over here, where I sort of divided the value of the neurons here by the healthy side to see if we see a reduction. In individual animals, we do see quite a few that do have a drop. where when they were exposed to TM230, those neurons died back, but we don't see overall patterns across the entire group. So this is a little bit inconclusive still. However, we still need to study the substantia nigra, which is the other part of that pathway, to check if there's neuron loss in that area as well. So we're still studying that. The other thing we wanted to look at was neuroinflammation, right? So neuroinflammation is a big part of Parkinson's disease. It's one of the hallmarks of PD, and we wanted to check to see if the immune cells of the brain were responding to the TMM230 and if that was potentially causing damage. So we stained for IBA1. This is just a marker of microglia, which are those resident immune cells of the brain. Again, we're looking in the striatum and the substantia nigra, which are those two regions that we are interested in Parkinson's disease. We're looking in the unaffected and affected sides. Visually, in these images, you can't really appreciate a lot of difference. It doesn't seem like there's a very strong microglial response. And again, that translates to when we graph this out and look at the data overall, we don't see a strong increase in the affected side across most animals. Although in this dark purple group here, which is one of the TM230 mutations, a couple of these animals like this one and this one have much higher microglia in the affected side versus the unaffected. So there could be individual cases where we have neuroinflammation, but it's not a strong pattern overall. So we're continuing to look at more markers. So beyond microglia, we're also going to look at astrocytes and other cell types in the brain to see if we can pull out some other patterns there. Okay, and then another big part of this is the reason we like to do animal models is because it helps us test if there's motor dysfunction, right? So Parkinson's disease is still defined in the clinic, at least, by the motor dysfunction. We want to see if our animal models recapitulate that motor dysfunction. So how do we test an animal for a PD-like phenotype? There's a couple of different ways. The two ways I used were catwalk and the cylinder test. So the catwalk is a long runway illuminated by a red light that has a green light running underneath it, so that when the animal crosses across the catwalk, we can capture its footprints. So if, in this video here, we're going to see an animal move across. You're going to see those little green paw prints that went across. The computer will capture those paw prints and map them out, and then we can measure a lot of things about its gait, like its stride length, its swing, its speed, things that we would see deficits in human patients. We can kind of match the equivalent in these animals. The other thing we're going to do is a cylinder test, which is a very easy test. We just put the animal in a cylinder and we measure paw preference when it makes a rearing behavior. Animals are not right and left-handed, so they don't naturally have a side they prefer. So when they have some kind of motor deficit, they should show a decreased preference for one side. So when these animals have motor dysfunction, there's changes in these tests, which we can quantify and then kind of assess. So here is the summary slide of all the data that I analyzed. So this is animals assessed after 12 weeks of being treated with that TM230. And this is the table summary coming out from that catwalk graph. So overall, we looked at a lot of different parameters of gait dysfunction. We found very few statistically significant changes. What we did find was a decrease in print area and max contact area as some of these animals were walking across that runway. So what that means is we're capturing the area of the paw print as they're stepping down on that glass plate, and changes in the area can indicate that they're stepping and walking a little bit differently, maybe a little bit more tenderly on their feet. So that could be a slight indication that there's a little bit of motor dysfunction in these animals. And then in that cylinder test, we don't see statistically significant decreases in paw preference, although in a couple of animals, we do have a drop in one side that they're hesitant to use. So again, small indication that there might be motor dysfunction, but nothing very large. And with that, I'm going to move on to some closing thoughts. So big picture, we've seen a little bit of work done in this TM230 model. The reason we want to study genetic models is because replicating genetic changes in the lab that cause PD, we can study how the disease develops based on those genetic pathways. By adding mutant TM230 cells specifically, we can observe how they react to it, which could give us clues as to how these mutations are contributing to Parkinson's disease in the first place. And overall, ultimately, we really want to evaluate whether and how these new TM230 mutations are causing Parkinson's disease, because that could help us learn a little bit more about the disease and potentially contribute to treating PD overall. And with that, I think this is my last slide. I don't have an acknowledgement slide, but I would be happy to take any questions. We can go back over any of the slides if we would like. Yes.
On your statistical significance, I don't see a great deal of significance. So what is that telling you?
What that's telling me, pardon me, what that's likely telling us is the behaviors of the deficits we're seeing in the model are very subtle, and they're not yeah, obviously they're probably not there, or they're not pulling out a variation, because animals naturally have a lot of variation. It's hard to get significance that pulls out above sort of the natural changes in how animals are moving. So we're moving into some high throughput cell screening assays with TM230 to see if we can do this in cell cultures with more replicates, to see if we can do sort of closer, more detailed studies to kind of pull out statistical significance there. But overall, yes, the animal model doesn't seem like we have a very strong phenotype for PD.
So the validity marker then is low.
Yes.
And at what point do you say, okay, this is not going to be a helpful study, I need to progress on to something else?
Right. So let's see. If we had gotten, say, no statistical significance anywhere, at that point we might have said maybe we should check something else. But we are seeing some patterns that do pull out as significant, and so there is something going on there. It's just kind of below, kind of right hovering around that region that we're able to detect with our statistical models. TM230 itself is a very promising candidate for Parkinson's disease based on the clinical and the genetic data in humans, because we see so many, relatively so many, it's a very rare mutation, but because we see multiple families around the world that have these mutations that get Parkinson's disease, it does seem like it is contributing. We just really have to find a way to see how. Any other questions? Okay.
Fantastic. Thank you.